Welcome back, everyone. I hope you all had a refreshing lunch break. We will now continue with our next keynote session. So it is my pleasure to invite Professor Siva Umapathy from Gyroid Photonics. Professor Umapathy is an honorary professor at IIC and former director of IISER Bhopal, and he's also renowned for his pioneering work in laser Raman spectroscopy and AI-driven spectroscopic methods, and is a recipient of several prestigious national scientific honors. We are delighted to have him today. Please help me call Prof. Umapathy to the stage.
Thank you very much. Oh my God. Thank you for the invitation, Sashi, and it's a pleasure to be here. I think it's the first time I'm giving a talk to non-scientific people. I mean engineering. So, it may sound too scientific, so forgive me for that. So I think most of you probably haven't seen me around. I have been here since 1990 in this campus. I've seen a lot, and I came back in 23 back to the campus after being an administrator for some time, and then retired in 24. And I still continue as an honorary professor, so I have my research group and students still in the institute. Like Nagaran said, I took the plunge into startup because I tell you, see, the thing is when you have patents and publications and experience, and one of the discussion I had is to see how do you now convert these patents into something commercial. And when the patents get extremely technical, only person who worked on it can actually convert it into commercial, because even the patent is difficult to understand for non-experts. I think that's very important. What Nagaran said, when you are at a PhD level at the frontier of science, sometimes even the patents are extremely specialized, and therefore it is important for you to get in and then start doing this. So we started this company called Gyroid Photonics. It's a deep tech, started at IISc as at the incubation center. So it's a part of IISc list of companies. So today, what I'm going to do is I'm going to talk about, briefly tell you what the technology is. Then I'll give you an example of a work we did in 2014, 2015, which made us realize the importance of AI, deep learning, not AI, I would say, deep learning at that time. And then we slowly started getting it. So we were one of the first people in the world start employing artificial intelligence to light signals. And then you see what are the challenges. So once if you learn that, now you go into hardcore AI, then you suddenly realize there are a lot of issues related to data manipulation, data curation, and all that. Once if you do that, cross that line of AI delivery, then you understand the impact of technology. So I can give you examples of what we have done, some real life examples in the last 3 or 4 years. And then finally, I'll give you a couple of examples of the products which are ready to market, and you can see why it is useful to the market. So what we do is basically we use laser light, and interact with the material. Is this working? Yes. So you see this, let's say this is your material. This material could be any chemical. And you interact and the light then gets scattered. The scattered light is measured, and the light is actually measuring the atom positions. You can see how the carbon or hydrogen and oxygen, how they are connected. The energy between the carbon and oxygen is fixed depending on what is connected to this carbon and what is connected to this oxygen. So the frequency of this EO, or the vibrational energy, or the frequency of light that comes out from this CO is different from this CO, because this C has no more C here. So the electrons that are not taken from here to there, therefore this CO will be higher in frequency compared to this CO. So this is what my specialization is, trying to understand how you understand atom moving in real time. How do you calculate which atom and which bond is moving in what direction? That's what we did for the last 35 years. So what we did, then we started working on light penetrating into chemicals, right? So the idea here is the lasers gives you unique advantage because the bond energies that you're looking at is extremely small. So you can't use a white light. So you have to use a highly pure, highly frequency pure light, which is called the coherent light. And because of this purity, you can actually even reduce the size of the beam to half the wavelength of this light, which means if I have 500 nanometers, I can focus it to 200 nanometers. When you look at that number 200 nanometers, you can see immediately that in a biological system, typical cell is about 1 to 2 microns and sometimes 30 micron. But the smaller size is 1 micron. Cells you have nuclei, you have organelles, you have cytoplasms. They are all in nanometers, 100 nanometers, 200 nanometers. Today, in a white light microscope, doctors can only look at the cells and the shape of the cells. So many of your AI applications in imaging are related to morphology of cells. But what we look at is the chemical composition because we can look at extremely small sizes. This is something that I have built a lab. If any of you are interested, you can go and see. The lab is just behind Hoysala, one of the first lab in the world in India to set up 10 to the minus 15th of a second laser pulses. Those laser pulses basically oscillate at the same time as the vibration of a bond. So if a carbon-carbon bond vibrate once, it takes 5 femtoseconds. So if I do experiments in femtosecond timescales, I watch atoms move in real time. And that laser lab was built in 2005, 2007. So it is still there. You're welcome to go and see that lab. And so when we are doing all these, so we learned about how to put light into a container and see what is inside. Therefore, I'm able to now look at chemicals either alone or mixed, right? So by looking at the signals that's coming out, when I put a light into a chemical like this, you'll have various frequencies coming. Of course, you have lambda 1 here. Same frequency also can come out. In addition, there are other frequencies which are coming. So I measure those frequencies. By then analyzing those frequencies, I can tell you what this chemical is. And that's what we did for the last 30, 35 years. So it's all led to a number of patents. But more importantly, the status today is we can penetrate actually up to 12 centimeter. So what we did to prove this, we actually bought a chicken tissue and then drilled a hole asymmetrically into a corner and heated a 1 centimeter diameter ammonium nitrate pellet. And then we scanned it at 100 micron resolution vertically and 1 degree resolution horizontally. And then we do a back projection algorithm imaging, and we can tell you exactly where the pellet is, what is the size, and where it is located. So this is something is very unique in the world, which means that you can do an imaging on 3 dimensions in a chemical specific way. What is the advantage? You can imagine if somebody has a cancer and you want to image their hand, and if you want to know where it is and what is the size, we can actually do that. So one of the ambitions that I always think, I don't know whether it will happen in my lifetime. Right now, if you go to a hospital, what you get is an MRI machine where you lie down, they scan the whole body and tell you the image, whether it is brain images or leg images. They only give you densities. All technologies that is available in the hospital today are density-based technology imaging. So this is the chemical-specific imaging. So what happens in our body is, as soon as a disease attacks our body, first thing that happens is the chemical changes starts happening. So if you can monitor the chemical changes in your body, you can predict diseases before you start becoming a symptom. And that's what we have been working on also in the last 2008 to 2025, that we look at body fluids, tissues, neurons. Those are the part of our portfolio of technologies where you can identify what the disease is, or are you going to get the disease. And this is the principle we use to do that. So what do we do? How do we do this? We actually ideally take a sample, look at this hyperspectral imaging or large spectral window, 2-dimensional, 3-dimensional spectra, convert them into curated data, put them into AI-based deep learning or some model, and then we actually do a visualization. This is a basic principle. This is a summary of what we do. But what is the advantage of a technology, something like this? So if you look at that, we can detect accurately what the chemical is. Whether it is inside, outside, doesn't matter. Why is it important? For example, in pharmaceutical company, Different companies make different paracetamol for you, whether it is Dolo, whether it is Crozin. They're all paracetamol, 500 milligram, but they are mixed with different additives. So you can identify which pharmaceutical belongs to which company because they use the same formula every time. So therefore, the only counterfeit chemicals can be identified. Similarly, you can imagine large number of applications in FMCG. Any company which are making products where chemicals arrive, they have to convert that into making sure that if you want to make the same product again, the raw material should be the same. So they can take that, petrol adulteration, diamond, jewelry changes, anything that is a chemical, you want to see small differences, we can look at them. And dangerous goods. So this is what we have done at Bangalore Airport. For example, somebody was carrying a paint thinner, which you are not supposed to carry. It is equivalent to petrol because it can it's flammable. So things like that. Not only that, but also lighter fluids, many dangerous chemicals. And then it's also non-destructive. Because we are now interacting with the laser light, the sample doesn't get spoiled. You can keep them. The reason it is important to be non-destructive, many a times if a doctor wants to look at the sample again after 3 weeks, you can go back and look at the data. Or in a legal system where the judge wants to see whether the same sample is not cheated, you still have the same sample, you can look at the data again. And minimal false positives. Because the chemicals are so different from each other, it's very rarely you get wrong information from the spectrum that you get. And of course, mixtures also can be identified. If you have two chemicals mixed up, by looking at unique features, we can select them out. And of course, no need to prepare samples. Time takes is about 2 to 5 seconds is where you see the results on the screen. And if necessary, when there is something dangerous chemical, for example, a corrosive or something else, and you don't want to go touch it, so you can even fire a laser beam and then measure the signal. And that's the advantage of what it is. So I'm going to show you an example of the kind of problem that people face when they start working on it. In 2012, 2014, one of the issues that people are talking about is hospital-acquired infection. These are the kind of different kinds of bacteria. It's called the escape, ESKPE. So escape pathogens, right? If you go to any hospitals, doctor will recognize that. If you have just had an operation, if you're in ICU, sometimes what happens after 3-4 days when you're trying to recover, because the wound is very fresh and you are immunocompromised, you may pick up one of these and then you will get into high level of infection. And the doctors can't identify whether infection is acquired in the hospital or elsewhere. So you need to know the source of the viral infection. So if you look at this, sample they use is either urine, blood, or the wound in which they found infection. So they take these samples and then do some analysis. So the way they do it is they grow the culture. And so what we did, we took exactly the same set of pathogen and tried to see whether we can identify which pathogen it is. If you look at the signatures of these different kind of bacteria, they all look very similar. So it's very difficult to identify from a naked eye. You can't do simple pattern recognition or even simple machine learning. We tried to do that. If you try to do that, you can see PCA in the simple machine learning. You can separate if it is only 2. When there is more than 3, you start having overlaps. And of course, when you have larger numbers, it's almost impossible to identify which belongs to which category. This problem we came across in 2013-14. We had to think about how are we going to solve this problem. And that's how we entered into deep learning. So then decided to take a ResNet model and try to apply this into the ResNet deep learning model. And we were able to get reasonably very high accuracy and actually close to 0.991, which is the close to 1 approximation. Now that we know we can do this, then we decided to use this model to an unknown set of samples. So blinded samples from the hospital, and that's what we did. And you can see here that we can actually get up to 98.66% accuracy of the unknown. So this is the ROC curve, sensitivity versus specificity, and this is what doctors like to see. When you get a new technology, you go show them, they'll say, where is the ROC curve? Once if we show them, they're happy that it works. And so this is the starting of our AI journey into application of AI into laser scattered light. So I decided, let's integrate now photonics with the AI. The idea here is that you take the data, and the data is, if you look at the data, you have to pre-process the data, because the data that goes in for training all should not have any errors in the system. And that's a major problem in when you're doing this. That requires specific expertise. So we have developed various models, mathematical models, to curate the data. And that's something is our group is well known in the world for data processing. Because you get scattered signal, the scattered signals will contain not only this light that is coming in, but you also have outside light that are coming in. In addition, you have signals from the sample itself. You'll have emission signals, you'll have reflected light, you have the frequency of the same frequency will come by reflected in multiple directions. So you need to delineate these signals from the signal that you are interested in. And that's a major job in doing that. And then you have to choose the AI model that you want to use and train and look for accuracy and then continuously train. Once the model is identified, you can use the model, put the unknown data, and see the prediction. But this comes 99%, you know you're in the right direction. So what are the challenges? Typical spectra looks like this, right? You see the background, for example. So this is the normal spectra. So we have to do, there's something called cosmic ray removal. Detectors are, because when we have UV visible detectors which are extremely sensitive, you have high energy particles which comes and hits. So unless you have a Faraday cage, which 90% of the time we don't. So in public utility cases, you can't have a Faraday cage. So the light particles actually hit the detector. You actually get unnecessary signal. So we have to do the analysis and remove those. And then we have to, the signals that the noise is, high frequency noise is too high. We have to reduce them. And the signals, for example, you have undulations of different kinds, you have to make them uniform. Because if you're going to give a set of data for AI to train, different set of data should have very similar architecture. And that's what we try to do. In addition to that, in terms of information that is useful for identifying the chemical is the frequency position. Which frequency are you looking at? The amplitude at each frequency, and of course the relative amplitudes between two frequencies, plus also the bandwidth and the peak shift. For example, one chemical will have slightly shifted from the other one. So therefore, the small chemical differences can induce small changes in any of these. So we have to train the AI to recognize these differences, and that's what we do when we try to develop the model. And so, how do we validate the model? What we do is, so this is an example for you. If you look at this escape pathogen that we discussed before, we use convolutional neural network, because if it's a non-linear data extrapolation, and the only way we should visualize it is something called stochastic neighbor embedding technology, where you actually convert that you plot the distribution of stochastic neighborhood distribution embedding, and you can see that they can be isolated. It gives an impression that you know that you can actually take them and then clearly do what you want to do. So you can identify each of the species separated after the AI modeling. And so we decided to now solve a problem. So during 2021, one of the issues all of us experienced is COVID. So the problem at that time was, during October, November, you not only had COVID, you also had flu. Because people are so paranoid with COVID, even a normal flu patient were actually treated like as if they had COVID patient. Therefore, the AIIMS director told me that, is there any way that can you develop a quick technology whereby as soon as the patient arrives, can we quickly separate them whether they are COVID patient or a flu patient? So we actually tried to develop a method whereby we can take these are all patients with similar symptoms when they arrive at the hospital. So we need to say, how can we diagnose, separate them out, and therefore the treatment of each of them. For example, COVID-19 is extremely contagious and flu is not so much, but the treatment for these are very different. So we have to somehow do something. So what we decided, we will actually look at this. Can we first start with COVID and flu, and then start indicating whether you have dengue or not. Now, the traditional technology, the gold standard that all of you know is RT-PCR. The advantage is that it's highly sensitive, small changes, but then you need to trained biological person to run them, and turnaround time is minimum 3 hours. It can be 24 hours, and of course, higher cost per test. So you need something that is simple, quick, within 5-10 minutes. As soon as the patient arrives, you can measure and tell them that this can be done. And the other one is antibody test, but it's not always the best way to do that. So what we did was we have been working on blood-related samples for the last 15 years. So therefore, we know that one of the ways in which we can look at infection is look at the blood signals. So we actually collected aims from Bhopal samples. They sent us, these are all work done in IISc. So we collected the signal from healthy control, flu, COVID, and dengue. We recorded spectra from all these patients. This is the typical data that you get from a light scattering signals. So then we actually look at the kind of symptoms they have. You have fever, cough, and you can see some people have fever. Percentage of people you have, 74%, first day only 7% has fever, but then they will have headache, cold, and cough. But then as the next day, you can see the percentage goes up, and then it comes down. So you can also monitor the age-wise. You can also monitor by type of, for example, this upper respiratory tract infection, lower respiratory tract infection. And the difference between flu and COVID is very, very small. They all have very similar kind of symptoms. And therefore, we had to somehow identify how to do this. So, of course, you can see typical machine learning. You don't get anything. Everything gets messed up. We only have an accuracy of 55. Then that time, since the data is so close to each other, because both are infection, then we decided to go to generative adversarial network model, where you can actually data generates back a similar kind of spectra to convert back and say, can you actually, can you continuously make another fake positive data and put a discriminator so that you can reduce the loss function during this whole training process so that you develop a model which comes here. And then you can use this to actually to get the final result of your AI. So what we did, we applied a stepwise model, which means if we take any two pairs, so you can see here what we do, we start with one test set, and then we separate them into healthy, control, and disease. And then the disease then separated into flu and COVID versus dengue, because these two are very close compared to dengue. And then you can then go and identify that, separate them. And you can see that after doing this model, this is the confusion matrix. You can see here the flu was 93% accuracy, COVID was 100%, and dengue was 100%. And this is something now we are now developing a technology to be implemented at the company. So if we do 3 microliters of the sample, and you do a laser spectrum, process it, the result comes out in 15 minutes from the time the patient arrives. You can separate them out into various groups of the patient, where the patient is healthy, patient is flu or COVID or dengue. So no sample preparation. Turnaround time is 20 minutes from the time you arrive. You take 5 minutes to take the sample, process, put it in the system, press a button, you get the result. So no need special laboratory conditions, because the blood is not a contagious sample. So I'm going to give you a couple of examples of technology that we are now going into market. And one of the technology, the basic principles that we have is, as I said before, body fluids, whether it is blood, whether it is urine, mucus, tears, all contain chemicals. So by looking at these fluids, we can actually tell you whether the disease is oncoming, or you already have the disease, or how serious it is. And that's the kind of research we have been doing by collecting data. Now, as you can imagine, our genomes are very different, right? If you look at South Indians, Kerala, Karnataka, Bangalore, Tamil Nadu, with the level of Dravidian genome versus Aryan genome mixed up. And then you go to northern part, you have Jains. So India itself is a very diverse country in terms of genomes. The genomes actually dictate the chemicals as well. Therefore, if I'm developing an AI model, which is a disease model that has to be applied to all Indians, then I need to have the diversity of blood samples. And therefore, the differences is going to be extremely small when you're training the AI model. So that's what we have done. We take thousands of samples and look at the unique signatures using AI, because the variables that you cannot physically see using machine learning can be identified by the AI. And that's what we try to do. So one of the things we did was a sepsis technology, right? So this was something we started very early with animal models. Using animal models, we figured out that it is possible to identify sepsis within 15 minutes from the time a patient arrives. What is sepsis? Sepsis is basically an advanced form of infection where your body organs are beginning to fail. Because the antibodies that creates in your body to fight the infection, when the concentration of the antibody goes up, they don't recognize which is your infection. It start attacking your lungs, it start attacking kidney, and therefore you go to a hospital, doctor will suspect that it may be sepsis. And that's what we do. So this is the problem all over the world. It's not only in India, the whole world is worried about sepsis. So that's the reason why we did. So we had a few patents on this. The idea here is very simple. We actually take a sample from patients in ICU, and we interact with a laser beam. This is the instrument that you see on the screen. You open this, you put the sample inside, close the door, connect this to a laptop, and then you run the experiment. You get a report on the screen which says that sepsis, no sepsis, or probable sepsis. And now this has been certified already by Ministry of Health. Now we are going to go for validation in various hospitals. So this has enormous impact in the healthcare industry, because the existing methods takes 24 to 48 hours. And of course, ease of use, you're not sampling anything, low cost, and of course, prognosis, reduced time in ICU. The other technology we have is this, which is what already been trialed at Bangalore airport. If anybody has a suitcase, when they find a chemical or something like this, they have to use this technology where they open the bag, take the chemical, and then do this chemical test by mixing chemicals. Then only they will know whether it is explosive or a drug or something else. In our case, we don't have anything, it's just a probe. That probe can actually go on without having to open this packet, it can look at inside. For example, this is what we have done already in my lab. We have looked at color bottles, plastic bottles, the envelopes. We can see what is inside without opening. So what you're seeing here, these bottles, are what we found in passengers' bags at Bangalore airport. And this is what people carry. So we can see whether it is, the guy will say this is coconut oil, and you end up finding it is actually thinner, the paint thinner, and things like that. This could be petrol. So this is milk. I still don't know why they was carrying milk in a checked-in baggage, but that's what we detected. So that's the kind of application. This is the unit we tested at Bangalore airport. This is the probe, and you can see there is a baggage ID scanner, and you have a printer, and you have a laptop which is connected to the machine. In the laptop, it will come and tell you dangerous goods or suspicious. What is the advantage of AI here? In this particular application, we have trained the AI to recognize the family of property of chemical. When I say the family, it will tell you whether the chemical is suspicious to be an explosive or not, or suspicious to be a narcotic. If it is fentanyl, there could be a number of fentanyls available in the world. When they make a new fentanyl, our system will come and tell you suspicious to be a fentanyl family. And so maybe in another 6 months, you might see this machine at Bangalore airport. Then you know it's from IISc. And we have other units that are upcoming, which is a backpack unit on robots and handheld and all that. And I'll skip that. I want to thank the guys. This is our team. And these are the private security agencies came to visit us because they wanted to see whether they can buy this technology. And I have to thank our funding agency, of course, IISc. We started with Startup India and then funded by Idea Spring. Nagan and the team were kind enough to give us funds to go to the market. And of course, Navam Capital as well. And thank you very much for your attention.
Thank you, Professor Umapathy. Now it's time for 1 or 2 questions. So the whole idea is how, though we might have initially thought, right, how can we bring AI into spectroscopy or even the chemistry or chemical physics and different areas, right? It's a typical core science field, right? And here this is one of the applications. Okay. Okay, one question from there.
So in one of your application where you identify dengue, flu, so in those kind of application, false negative play major role. So like if you identify dengue patient as a flu, so it's a major health hazard for them.
Correct.
So how you address those kind of scenarios?
Well, there's a scientific answer and a commercial answer.
Scientific.
Scientific answer is that about 2 to 3% are false negatives, and so that's very difficult. I mean that can become smaller and smaller when the data gets bigger and bigger. So we have to wait until we get to that point. But the existing technology are much worse because we are at least close to 3%. Most of them are 15-20%.
Do you think any advancement in laser spectroscopy can make that difference in future?
No. I think we are already very advanced. What's going to make difference is that the number of samples and diversity in samples and better AI models, which can actually identify even extremely small percentage of variable differences. That can make a big difference.
Thanks.
Okay, if there are questions, maybe I have one small question. Yeah, somebody is here. Yeah, please.
Thank you. My name is Dr. Rajesh. That was a brilliant presentation and awesome inventions. Thank you so much. My question is about the Cinesco's approach when we project the AI-based health innovations. Was it easy because even the US FDA initially had challenges, but now they approved tons of AI-based applications. So what is your experience?
Yeah, so I think FDA itself, they themselves are very aware. If you're going to give it to a technology, replacing the existing technology, but still predict the same thing, they will not accept it. So if you have a technology which is new, challenging, and there is nothing available, I mean, this is only, for example, there are many cancer drugs, efficiency is only 30 to 50%. They still permit them because there is nothing else is available. So we are at that stage right now with the AI and diagnosis. And once if we reach a stage, as they say, there's always pitfalls. We don't know what will happen, but they will agree. Yeah.
Thank you.
Okay. If there are no more questions, then let's thank the speaker once again and for the wonderful talk. Thank you, Prof. Umapathy, as a token of appreciation. A small.
Okay, thank you.